Hello student, good afternoon. How are you all? Hope you are doing great. I am your biology mentor and today I am going to deal with the chapter Plant Kingdom. So let us start this session. So this is Biology for NEET Day 4 course and we are going to start the chapter Plant Kingdom. So this chapter we are going to study in two parts. The first part we are going to study about the basic introduction about the plant kingdom. Then we are going to study about different system of classification. Yes. So the first system that will be artificial. Then we will discuss about the natural system of classification, phylogenetic and also the phenetic type of classification. So in the phenetic type, we are having different types of taxonomy, that is numerical taxonomy, cytotaxonomy, chemotaxonomy, right? So that all we are going to study about. And then we will study about algae in great detail. We will study about the classification of algae that deals with the chlorophyceae, phaeophyceae and rhodophyceae. These are the three different classes of algae. So that all we are going to study. And also we will discuss about the ecological importance of algae in our daily life. So let us see this chapter with the eye of the NEET examination. Then this chapter is having some questions every NEET examination, right? So if we see the prior examination of the NEET, then we will find that every year 3 to 4 questions come from this chapter. So this is the weightage for the NEET examination for this particular chapter. Okay, so let's start the chapter. So we look at the broad classification of living organism under the system proposed by Whittaker. Yes, we have studied about the Whittaker classification in the biological classification chapter, right? So here, according to the Whittaker classification, it has been divided into 5 kingdoms, right? That is the Monera, Protista, Fungi, Animalia, and Plantae. So in the Monera, we are having the prokaryotic organism, right? And in Protista, eukaryotic unicellular organism, right? And we will have a different kingdom, that is of fungi, and the two other different kingdom, that is Animalia and Plantae. So here we are going to study about this Plantae, right? We are going to study about this Plantae. So that is why this chapter is called as Plant Kingdom, right? Plant Kingdom. Okay. So, what does the plant kingdom constitute? What type of plant this kingdom have? So, they are basically having the first type, that is algae. The second type, that is having bryophytes. And the third type, that is having pteridophytes. Fourth one is gymnosperms. And the fifth one, that is the flowering plant, that is angiosperms, right? So this algae is very different from the other 4 types, that is the bryophytes, pteridophyte, gymnosperm and angiosperm. So what is that? In which respect it is quite different from the other 4? The algae do not the plant cannot be differentiated into the true leaves, stem or roots, right? So this algae is not having true stem, leaves and roots. So their body is like thallus. Their body is like thallus. So that is why it is kept in thallophyta. Thallophyta, right? So let us discuss about algae in this chapter. Before going to that, we will have a discussion on the different types of classification. So the first one is the artificial system of classification, right? So it is based on 1 or 2 morphological characters for grouping organism together. So what we do basically in the classification, we generally place some of the organism that is having a same type of characteristic we group in one group, right? And the different type of organism in the different group based on their characteristics. So we are basically looking what? We are basically looking for the characteristic. And the common characteristic organism we are keeping in one class or one group, we can say. So for this artificial system of classification, we are just considering the morphological character. Morphological character means the character which we can see from the eyes, right? We are not looking the anatomical structure, we are not looking the behavioral structure, right? We are just looking the outer structure of the plants. So that is why this particular classification is also called as artificial system. This is not a true system of classification, right? So some artificial system have used habit and habitat for this purpose. They have just considered the habit of the organism. They have just considered the habitat of the organism. So that is not a good for a basis of the classification, we can say. So let us talk about some of the example who has followed this particular type of classification. Aristotle classified animals on the basis of their habit: aquatic, terrestrial, and aerial. So if we talk about the aquatic, then we will keep fish in this, right? If we talk about the terrestrial, then we can say reptiles, right? And if we talk about the aerials, then we can say birds, and also bat. That belongs to the mammal actually. So this is not a correct way of classification. We will discuss about the drawback of this classification. Pliny also distinguished animals on the basis of flight and non-flight. So superficial characters they are considering, right? Linnaeus classified organism on the basis of numerical strength of sex organs like androecium, number in flowers of a plant. So only the number of androecium Linnaeus considered for the classification system, right? So this is also comes under the artificial system of classification. Now, let us discuss what about the drawback. Closely related organisms are separated into different groups. So, closely related organisms are separated if we are just considering the morphological characters. System is based on one or two morphological or ecological traits. We are just considering the morphological characters or the ecological traits. And in this system, what happened? The closely related organisms are kept far away, and dissimilar characters governing organisms comes very close together, right? So this is not a correct way of classification. Some other drawbacks: system picked up artificial characters for the classification is not able to reflect the natural relationship. Very important point. So this particular system of classification is not building the natural relationship amongst the organism. Do not show clear-cut evolutionary relationship. This is not even focusing the evolutionary relationship among the organism that belongs to a particular group, which is very, very important. Some character used in the artificial system of classification get changed with the change in the environment. For example, if a particular plant is growing at one place, right, and it is not getting proper nutrients, so it will have small leaf structure, small leaves. And if it is grown in an environment which is having a good nutrient supply from the soil, good water supply, then what will happen? They will have a proper growth of the roots, they will have proper leaf structure, large leaf structure, right? So if we are just considering the leaf structure for the classification, then this is not going to work out, right? So some character used in this artificial system classification got changed with the change in the environment, and if we are considering that particular character for the classification basis, then that will not going to work out. Okay, so next comes the natural system of classification. Very interesting. In this system, number of character into consideration that brings out natural similarities and dissimilarities. So here they are considering the natural similarities, and also they are considering the dissimilarities among the organism. So this system employs the comparatively constant character that is not going to change with the environment, right? So this particular system is quite acceptable because they are considering the natural similarities among the organism and also the dissimilarities. They are not only considering one or two morphological characteristics, right? So they are considering the number of characteristics. It includes morphological characters. Yes, just now I said that they are considering the number of characteristics. So that could be based on the morphological characters, that could be based on the anatomical characters. Internal structure also they are considering for this, right? Morphological characters means they are considering the outer structure, how the plants look like, what is the leaf structure, what is the root structure, what type of the stem they are having, how does the flower look like, how the fruit is. So all these comes under the morphological characters. Now, anatomical structure. They are considering the anatomy means the tissue level of structure. Cytological characters, they are also considering the cytological character, means the characteristic that is based on the cellular structure. Physiology, cytochemistry, and also the reproduction. So number of things they are considering in the natural system of classification, and that is why this system of classification is accepted. Now let us talk about the merit of the natural system of classification. It brings out affinities on the basis of natural characters. Affinities based on the natural characters. So all the characters of a particular group, they are having a number of organisms. So all the organism will have that particular character, and they will having affinities with that. It placed only related organism in a group. So if they are having the affinities to that characters, then all the related organism will comes to that group. This system prevent coming together unrelated organisms. So we are not only considering the similarities of the character, but also we are considering the dissimilarities. We are not grouping those organisms which are dissimilar in their characteristic, dissimilar in the morphological characteristics, or dissimilar in the anatomical characteristic, or dissimilar in the cellular characteristic, etc. So we are considering all those number of characteristics, right? And also similar. If all these characters are present similar in two different organisms, then we will put these two different organisms in one group. And if they are not having any one single character, one organism is absent, then we will not place that particular organism in that group. Okay, so unrelated organism will not come in one group. So this indicates phylogenetic relationship and the origin of the taxa. It also talks about the phylogenetic relationship, evolutionary relationship. We can say that amphibians has been developed from the fishes like organism, right? So they are considering that thing also. Classification, phylogenetic system of classification. classification is based on the evolutionary relationship of organism is called as phylogenetic system of classification. So in this particular system of classification, we are just not only considering the different characteristic based on the morphology, based on the anatomical structure, based on the cytological structure or physiology, but also we are considering the evolutionary relationship among the organism, right? So that talks about the phylogenetic system of classification. If you are considering the evolution, means you are discussing that this particular organism has been developed from this particular organism or this dead organism which is form which is recently we got from the fossil, and it is showing the same characters. So this shows that this present organism has been developed from this fossil organism. So if we are discussing such type of thing, then we are talking about the phylogenetic system of classification, and we are focusing evolutionary relationship among the organisms. Fossil, just now I talked about. Fossil play a very important role in elucidation of the evolutionary relationship, because there are so many organisms which we are not having present time, but yes, they were flourished during some previous era, right? Like dinosaurs, they were the biggest reptiles in a Jurassic era. So now they are not present, but they are present. They are found in the form of the fossils, and they are the link that develop the evolutionary relationship among the organisms which are present today and in the remote past. So the fossils play a very important role in the evolutionary relationship and also developing the phylogenetic system of classification. So it is based on the possible evolution of different traits. Yes, different characters that are followed for the evolution study of the evolution of organism. So those traits are followed for the study of the phylogenetic system of classification. And yes, it talks about the evolution of organisms. Phenetic classification, another type. This system of classification is based on the affinities, similarities, dissimilarities of the characteristic present in the present day organism, without searching for the evolution and diversification of these traits in their fossil ancestors. So we are not considering evolution or the fossil ancestors. But yes, we are considering the affinities, similarities, and dissimilarities of the characters which are present in today's organism, the organism that are present today. But we are not considering, we are not searching for the evolution. We are not talking about the how this organism has been evolved. We are not talking about that. So that is the phenetic classification. So this phenetic classification has 4 branches. So the 4 thing that we are considering for this phenetic classification, that is 4 different types of taxonomy. The first one is cytotaxonomy. The second one is chemotaxonomy. The third one is numerical, and the fourth is cladistic. So all these comes under the phenetic classification system. So let us discuss about one by one. Cytotaxonomy. Classification is based on the information provided by the comparative cytological studies. We are studying the cellular structure, but how? We are comparing different cytology. Suppose we are studying about the prokaryotic and eukaryotic cell. So prokaryotic cell cellular structure is very much different from the eukaryotic structure. Chromosome structure. Prokaryotic cell is having just single chromosome, whereas the eukaryotic, in the nucleus, they are having different number of chromosome, and their chromosome number is fixed according to the species. We are also discussing about the different structures, cellular structure, ribosomes. Ribosomes in the case of prokaryotic, it is 70S type, whereas in the case of 80S, it is eukaryote, it is 80S type, right? So these are the things we are considering in the cytotaxonomy. We are also considering the meiotic behavior of the chromosome, how the meiotic division takes place. This system of classification is based on the characteristic of the various chemical constituents of the organism like amino acid, proteins, DNA, sequences, and alkaloids also. So cytotaxonomy deals with the chemical constituents of the organism. What are the chemical constituents of the organism? We will deal about the amino acid. We will deal about the proteins, specific proteins. We will deal about the DNA sequences. That is, every species is having different DNA sequences. We will also deal about the alkaloids with that, and that are very specific to the species. So how we are going to consider this? We will going to consider when we will going to study about the cellular structure, right? Numerical. So it evaluates the resemblance and the differences or the primitiveness and the advancement through the statistical method based on the number of characters, right? We are going to study about the number of chromosome. Then this is the numerical taxonomy, right? Number of pollen grains, numerical taxonomy. So these things, when you are building a statistical data based on the number of characters, then you are following the numerical taxonomy. And these are the very specific things that we are going to consider for the classification basis. And it is so specific, then you will have a similar type of organism in one group. See, they are so specific. It is easily carried out using the computers. Is based on observable characteristics. Number and codes are assigned to all the characters and the data are then processed. So all these numerical data are feed up in the computers, right? And then we process that data and we will get some statistical results. And on the basis of that, we can develop some code. And from that code, we can classify the organism. If the similar codes are having, then all the organisms that are having the similar code, that will be put in one group. So what we are considering for the numerical taxonomy? We are considering the numbers. It is all the place of the numbers, the number of chromosome, number of sequences based on chromosome, right? All these things can be considered, fed up in computers in a database. That database will be used for some statistical results. That statistical result will give you some codes, and that code will be used for taxonomy purpose. So I hope this could be easy to understand, that what are the cytological structures, what are the cytological structures, what are the numerical structures that we are considering for this type of classification. And the fourth thing that we are considering for this, that is cladistic. It searches similarity due to the common phylogeny or origin from common ancestors. So what is phylogeny? So the phylogeny is the history of the evolution of a species or a group. It talks about the history. It talks about the history of the evolution. History of the evolution of a species, of a particular species or group. especially in the reference to the lines of descent and relationship among the broad group of organism. So such tree-like structure we can have like this, right? So here will be one organism, suppose it is A, then from this B, C, D and E, right? So all these organism has been developed, right? And it is not a short duration of time, but yes, it is a very long duration of time, and we can say that this organism A evolved to different organism B, organism C, organism D and E. So here we are talking about the history of the evolution of one particular species or a group of species, right? So there are two types of characters: ancestral characters or derived characters. The ancestral characters means the characters that we are today also we are having some of the characters that came from our ancestors, and over a course of evolution we have derived some of the characters, right? So that are called as derived characters. Okay, so it is clear, hope it is clear. So that is the cladistic. Kingdom Plantae include all eukaryotic chlorophyll containing organism commonly called as plants, and that is why on the basis of this eukaryotic chlorophyll containing organism all these organisms are kept together. uh, Algae, bryophytes, pteridophytes, gymnosperm, angiosperm, all these are eukaryotic and chlorophyll containing organism. So they are all kept in one kingdom that is called as plant kingdom. This is the best example for the cladistic. Plantae includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. So all these are the eukaryotic chlorophyll containing organism. Thallophyta. So I have already talked about the thallophyta in the beginning of this session. So it comprises the simplest plant which possesses undifferentiated or a thallus form. So if you see the structure of any algae, then you can see they can be in a filament form, they can be in a cellular form, they can be in a colonial form. Different cells unite together and form a colony. But you cannot say that this is the structure that shows the leaf like, or this is the structure that shows like a root, and this is a stem like structure. We cannot say in the case of algae. So it is their structure, their body is like thallus. Reproductive organ single celled, non-jacketed and called gametangia. So this is also one of the very important characteristic of thallophyta, means this their reproductive organ of algae, they are non-jacketed and they are called as gametangia. Reproductive or organs are called as gametangia, and they are not covered by any jacket type of cells. So this we can see in the case of algae, and that is why it is also called as kept in thallophyta in another type of classification. Their embryo stage, vascular and mechanical tissue are all absent. We do not find any type of embryonal stage in the case of algae. We do not find any type of vascular structure in algae. No mechanical tissue in algae. So all these structure are totally absent in the case of algae. Asexual reproduction by accessory spores is very common. So we are having different types of spores formation in the asexual mode of reproduction in thallophyta, and presently it includes only algae. So basically we are talking about these characters belongs to the algae, and they are kept in one type of classification, that is in thallophyta. And this thallus, from how this thallophyta name originated, from the thallus like structure, because their body is just thallus, ribbon shape, ribbon shape. No true leaves, no true stem, no true root-like structure. Right? So that is why it is called as thallus. Their body is like thallus and it is kept in thallophyta. So these are the general characteristics of thallophyta. So let us start first organism, a type of organism for the plant kingdom, that is algae. Algae are simple thyroid chlorophyll containing autotrophs. So they are autotrophs. Autotrophs means they can make their own food. Why they can make their own food? Because they contain chlorophyll, right? And they are thyroid structure. Both freshwater and marine habitat. So they are found in freshwater also and also in the marine environment. All also seen in other habitat like moist stones, soils and wood also. So they are not only present in the aquatic environment, but they are also present on the moist stones. The stone which are moist, which are having a film of water. Soil, wet soil, they also contain algae. And sometimes you can see on the bark of wood that is quite wet, especially if you go to the rainforest area where the wood are always wet, and on the surface of these wood you can see algae growing on that, right? So they are also seen in such habitat like moist stones, soil and wood. Some of them are associated to the lichens and on the animal body like sloth, bear. So first of all, let us discuss about the lichen. What are lichen? Lichen are the special group of organism. They form this lichen structure and by combination of two different organism, algae and fungi. You have studied this about in the second chapter, biological classification, right? Lichen. So they are having the two component, algae and fungi, phycobiont and mycobiont. So the algae there do the process of photosynthesis and provide food to the fungi. And what does the fungi do? Fungi absorb water and mineral from the substratum and provide to algae, because without water there will be no photosynthesis, right? So algae do the process of photosynthesis and provide food to fungi. So this is the mutual relationship, we can say. Sloth bear are generally wet, so their hairs are having some growth of algae. So on the animal body also we can see these algae growing. The size and the form of algae body, microscopic to unicellular form like the chlamydomonas. They are microscopic. You cannot see the chlamydomonas with your naked eye. You need an aid, that is microscope, and they are unicellular alga. Colonial form like volvox and filamentous form like Spirogyra and Eulothrix. So let us see the figure. Here you can see the unicellular flagellated, that is the chlamydomonas, single cell, right? And here one more unicellular microscopic, that is chlorella, but they do not have any flagella-like structure. So they are non-motile. Filamentous form, Spirogyra. You can see the filament of the Spirogyra here. And they are having a spiral chloroplast. Colonial form, volvox. You can see the colony. All these green dot-like structure are the single cell. And you can see the number of cell they are having. And in fact, they are having some daughter colony also. And this one is also called as the parent colony. Right? So this is the colonial form, that is example is the volvox. Branched filamentous, ectocarpus and parenchymatous structure also we can see in the form of ulva. So you can see they are having different form of body structure: unicellular, microscopic, filamentous, colonial form, branched filamentous and also parenchymatous type. Certain marine form algae assumes a massive form, that is also called as kelp. So you can see this is an alga that is called as kelp. A typical alga cell shows a cell wall enclosing organelles like endoplasmic reticulum, Golgi bodies, mitochondria, ribosome, chloroplast, vacuoles, and nucleus. So all these basic cell organelles are present in alga. And the one which is very keep this particular organism in plant kingdom, that is presence of chloroplast and also cell wall. Special storage bodies. This is very important. This characteristic is very important. Number of times on this characteristic question has been asked in the NEET. So special storage bodies, that is called as pyrenoids, are present in the chloroplast of green alga forms. In the green alga, they are having a special storage bodies. They store starch, which is called as pyrenoids. It consists of core protein surrounded by starch sheath. So basically they are having in the center core protein structure and surrounding structure is the starch. And in center there is a core protein. So this is the pyrenoid structure that is present in the green alga, and this is considered as storage bodies. Pyrenoids are the organ of starch formation. Algae reproduce by vegetative, asexual, and sexual mode. So we are having all the three different types of reproduction that we can say: the vegetative, asexual, and sexual method. So let us see one by one. Vegetative propagation by the fragmentation take place in the favorable condition. So what happened? Suppose the spirogyra is there. This is the fragment of a spirogyra, and due to the water current, sometimes the water currents are so heavy that it breaks the spirogyra in 2 or 3 fragments, and all these fragments now behaves as a new individual. So this is a form of vegetative propagation, right? And this occur in a favorable condition. Asexual reproduction, zoospores, aplanospores, and echinids. These are the 3 different types of asexual reproduction. So zoospores are motile. Now here you can see zoospores. Why it is called zoo? Spore we can understand, but zoo word is attached here because they are having this flagella, and due to this flagella they can move from one place to another, right? So animals generally move from one place to another. So here it got its name zoo from this particular characteristic, because this particular spore is motile. So they are motile. Aplanospores are non-motile. So here you can see aplanospores, and generally the aplanospores and these echinids are formed when the plant is going for the resting or unfavorable condition. When the condition is not favorable, so they form aplanospores or echinids, right? So they are a thick walled resting spores that is formed in wearing condition, means condition that is not favorable for the growth of algae. So what they do, they form aplanospores and echinids, and when the condition become favorable, these aplanospores and echinids germinate and give rise to new individual. Right? So these are the 3 different mode of asexual reproduction in the case of algae. Right? Sexual reproduction is possible by the fusion of gametes, and it may be isogamous, anisogamous, and oogamous. This is also very important point from the examination point of view. Isogamous, anisogamous, and oogamous type of sexual reproduction. So fusion of similar gametes with respect to their size and shape. So when the two gametes, they are motile, they are similar in structure, morphological and physiological also, both morphological and physiological, similar, and they unite together and form zygote. then this is called as isogamy. An example, it is found in Eulothrix and Chlamydomonas also, right? So these are the some of the example: Eulothrix, Chlamydomonas, and Spirogyra. Physiological and morphologically, both the gametes are similar, and that is why it is called as isogamy. Fusion of gamete dissimilar in size, morphologically dissimilar. So one is smaller and one is larger. So morphologically they are dissimilar, or sometimes they are morphologically similar but physiologically they are dissimilar. So this is called as anisogamy. An example is Eudorhina. Fusion of motile small male gamete with a large non-motile static. So here we can see oogamous type of sexual reproduction. Here this is the male which is motile. Motile means it is having flagella, so it can move. Here it is an egg that is non-motile. This is the female and this is non-motile. So when these two fuse together, sexual reproduction occur. But this is oogamous type. It can be seen in the volvox and fucus. So here if you see clearly this picture, isogamy, both the gametes are morphologically similar, and right, and here anisogamy, and here in the case of oogamy, one is larger and non-motile and one is smaller and motile. Classification of algae. Algae are usually differentiated on the basis of their pigments, flagellation and storage products. So if we discuss about the classification, then we are going to it can be divided into three different classes, that is chlorophyceae, phaeophyceae and rhodophyceae. So chlorophyceae, it is also called as green alga, and it is having chlorophyll A and B. This is very important. What type of chlorophyll the chlorophyceae is having, what type of chlorophyll the phaeophyceae is having, and what type of chlorophyll the rhodophyceae is having. So here chlorophyll A and B, that is present in the green algae. Chlorophyll A and C, and along with the fucoxanthin, another brown pigment that is found in the brown algae. Chlorophyll A and D, and along with the phycoerythrin, that is found in the red algae. Stores food material is starch. In the case of phaeophyceae, mannitol and laminarin, and in the case of rhodophyceae, Floridian starch. Cell wall is made up of cellulose. In the case of chlorophyceae, cellulose and algin. We will discuss about this in the coming slide. Very important, algin and cellulose. In the case of phaeophyceae, cellulose is having, in the case of rhodophyceae, cellulose, pectin and polysulfate esters. Their cell wall is made up of. Very different, right? Flagellar number and position of insertion 2 to 8 and equal and apical in position. 2 unequal and lateral. So the flagella will not be equal in size. Absent. No flagella. Very important point. Rhodophyceae do not have any flagellated cells in their life cycle. Very important point. Habitat is fresh water, brackish water and salt water. So they are present in fresh as well as salt water. Phaeophyceae, they are present in the fresh water and again salt water and brackish water. Rhodophyceae, they are present, some are present in the fresh water, but mostly they are present in the salt water or the brackish water. So let us discuss the first class, that is the chlorophyceae. The members of the chlorophyceae are commonly called as green algae because they are green in color and they are present chlorophyll A and B along in their plastids. Carotene and the xanthophyll are the accessory pigments that are also present in it. Majority of the fresh water, that is 90% of the individuals are fresh water. They are present in the fresh water, but some are present in the marine form also. So let us see the unicellular motile form, that is the Chlamydomonas, unicellular non-motile, that is Chlorella, and colonial form Volvox. Filamentous form, filamentous unbranched. You can see here, filamentous unbranched, that is in the case of Spirogyra and Eulothrix, both are unbranched, but highly branched with the main axis and lateral branches that we can see in the Chara. So we are having both the type, filamentous unbranched and filamentous branch type in the case of green algae. The cell wall consists of the inner cellulose and outer pectin or pectose layer. So the inner layer of the cell wall is made up of cellulose, and outer layer that is having the pectin, layer of pectin. Chara, the cell wall, shows the calcium carbonate deposition. Right? So Chara is, just now in the previous slide we have seen, they are having the branched filamentous type, and they are having lot of deposition of the calcium carbonate on their cell wall. The reserve food material is the starch. Their chloroplast is of different shape. So here you can see the cup shaped in the case of Chlamydomonas. This Chlamydomonas is having cup shaped. Girdle shaped in the case of Eulothrix. This is the Eulothrix. It is having the girdle shape, and discoid form in the Chara. So 3-4 more different form of the chloroplast we can see. Ribbed in the case of Volvox, reticulate in the case of Oidogonium. So here you can see the reticulate type, and spiral shape, like the spiral shape structure in the case of DNA we can see. So same type of the chloroplast structure that is found in the Spirogyra. Stelliate, star-shaped structure of chloroplast in the Zygnema, here you can see. So the different shape of the chloroplast we can see in the chlorophyce. Reproduction in chlorophyce: vegetative, asexual, and sexual method. All the 3 different types of sexual reproductions are found in the case of chlorophyce. Vegetative reproduction result by the fragmentation. We have already discussed this in the general characteristic, right? Asexual reproduction take place by the zoospores, aplanospores, and echinids, and these are the resting spores formed when the condition is not favorable for the growth of algae. Zoospores are formed during the favorable conditions, and aplanospores formed during the unfavorable conditions. Sexual reproduction occurs by isogamous, anisogamous, and oogamous type. We have already discussed what does this mean: isogamous, anisogamous, and oogamous. So Eulothrix and Spirogyra showing the isogamous mode of reproduction. This you have to understand and memorize, that what type of sexual reproduction is there and their examples, right? So isogamous example, Eulothrix and Spirogyra. Eudorhina, certain species of Chlamydomonas, shows the anisogamous mode of reproduction. Means their gametes are morphologically or physiologically dissimilar. Volvox and Fucus show the oogamous type of reproduction. So in this case, the female will have non-motile gamete and the male will have motile gametes. So you have to understand and memorize the examples of the 3 different types of sexual reproduction, right? Okay, so zoospores and the gametes are the biflagellated and motile. We have seen the zoospores, they are biflagellated and they are motile in structure. The fusion of gametes results zygote, which takes place a period of rest and later develops into a new algae by the cell division and growth. So this is just a cell that shows a diploid structure. Otherwise, all the cells which are formed in the cell cycle of algae are haploid in structure, right? Chlamydomonas, Volvox, Eudorhina, Eulothrix, Spirogyra, Cladophora, Chara are the common examples of green algae. Phaeophyce, second class, that is also known as brown algae. So brown algae results due to the pigments fucoxanthin, which marks the green color. So they are generally green in color because they are having the chlorophyll A and C. But they appear brown in color. Why? Because they are having a lots of brown pigment that is called as fucoxanthin. So it marks the green color due to the presence of chlorophyll A and C. And that is why it appears brown in color. Chromoplasts are ellipsoidal and discoid and contain chlorophyll A, chlorophyll C, carotene, xanthophyll and fucoxanthin. Most of the brown algae are marine and few are freshwater. We have already discussed all these points in the tabular form also. So generally they are attached to the rock with the discoid or the branched holdfast. So here we can see the three different structure, that is holdfast, stipe and frond. Holdfast is the structure that attach this brown algae to the substratum. Stipe is a filament like structure, and green leaf like structure that is also called as frond. So this is frond is showing the parenchymatous structure. So the thallus ranges from the simple to the complex parenchymatous form. Plant body show holdfast, stalk like stipe and the leaf like lamina that is also known as fronds. Lamina, fronds is a photosynthetic in nature. So this lamina, this is frond, this is also known as frond. They are doing the process of photosynthesis, right? Stipe and holdfast do not contain this chlorophyll. So they do not do the process of photosynthesis. Cell wall is differentiated into outer gelatinous layer, algin, very important point. So the cell wall can be differentiated into two form, means two layers are there. The outer gelatinous layer that is made up of algin, this is the hydrocolloids, and the inner layer that is made up of cellulose. Storage product, that is the laminarin and mannitol. Large forms often possesses air vesicles or bladders that provide buoyancy to the organism. So these algae can float on the surface because they are having air vesicles or bladders. Phycocolloids of the brown algae are non-sculptured and mucosaccharides. Some common alginic acid, fucoid and fusenes are also present. Phycocolloid protect brown algae from the desiccation during the low tide, freezing under the low temperature. So this is very, very important. These phycocolloids protect the brown algae from being desiccated when there is a low tide, when there is a less amount of water, and injury when the beaten against the rock. So these phytocolloids are very important for these brown algae. Vegetative, asexual and sexual method, all the three different mode of reproduction we can see in the Phaeophyceae. Fragmentation is the common method of vegetative multiplication. All brown algae reproduce asexually by the zoospores. So asexual method with the help of zoospores occur. So when the cell divide, it give rise to 4 zoospores by the process of meiosis. So all, suppose this is a cell, right? It undergo meiosis and it give rise to 4 zoospores. So asexual method of reproduction occur with the formation of zoospores. The zoospores are pyriform or the pear-shaped with 2 laterally attached unequal flagella. So basically they are having pyriform shape. One is larger flagella and one is smaller flagella. So this pear shape. Sexual reproduction take place by the isogamous, anisogamous and oogamous method, and gametes are more or less similar to the zoospores. In isogamous and anisogamous, both the gametes are motile and with the heterognate is flagellation. In oogamy, only the male male gamete is the flagellated, while the female is non-flagellated. We have already discussed in general characteristic of the algae all these things, right? In most algae forms, the fertilization is external, but it is internal in oogamous reproduction. So this is important point. Oogamous reproduction is showing the internal mode of reproduction, right? And because this egg structure, which is non-motile, it is attached to the thallus. So the male one is entering to the female thallus or the oogamous structure. egg structure, and that is the eugamous type of reproduction. Rhodophyceae, the third class, that is also known as red algae, due to the presence of coloring pigment R-phycoerythrin, which completely masks the chlorophyll A and D. So again, it is green in color, that is due to the presence of A and D, but it is having lots and lots of red color pigment, that is phycoerythrin, and due to that it imparts red in color. Majority of the members are marine and some live in brackish as well as freshwater, but majority are marine. Batrachospermum is the freshwater red alga. So this is the important point to remember. One freshwater alga, that is Batrachospermum. This is one of the example. And mostly the red alga are found in tropical seas and have filamentous form. Some members show the epiphytism and parasitism. So some of the red alga shows the parasites. They are parasites, and also they get attached to some other organism. So that is called as epiphytism. Sometimes they are attached to the snail's covering, right? Thallus of red alga ranges from simple to the complex multi-axial form. Cell wall is composed of cellulose, pectin and polysulfate esters. So they are having cellulose, pectin and polysulfate esters in their cell wall. Storage product is Floridian starch. Very important to remember. This Floridian starch is the food reserves. Structurally, the Floridian starch resembles to the glycogen and amylopectin compounds. Many alga forms have extracellular deposition of the calcium carbonate on their wall. So again, like fucus, we have seen cellular calcium carbonate deposition, right? So here many red alga also have this calcium carbonate deposition on their cellular structure, and it provides protection to them. Vegetative, sexual and sexual method we are having in the red alga also. Vegetative reproduction by the fragmentation process, and non-motile spore, very important. Carpospores, this is the carpospores, are the means of asexual reproduction. You have to just remember this name, carpospore, that shows the asexual mode of reproduction. Sexual reproduction is oogamous, and it is followed by the complicated post-fertilization changes in the zygote and results in the fruiting body. So this is the alga, that is the red alga that form the fruiting body, right? And this spore is quite different from the other two alga, that is carpospore. This occur only and only in the red alga. Red alga exhibit the alternation of generation, means it is showing the haploid structure as well as the diploid structure. Motile spores and the gametes are totally absent in the red alga. This is very important point, and many times it has been asked in the examination. Economic importance of algae. Phytoplanktons, so they are contributing major role for the process of photosynthesis in the aquatic medium, right? About half of the total photosynthesis that is carried by the entire algae. So whatever the oxygen we are getting today in the atmosphere, mostly we are getting by the algae, by their photosynthesis process. Common edible algae, some algae are edible, that is Porphyra, Laminaria and Sargassum. These are edible algae. Phycocolloids, that is algin, carrageenan, are commercial importance. They are used as a phycocolloid. They are extracted from the red and brown algae, because algin is found in the brown algae and carrageen is found in the red algae. The brown algae, Chondrus crispus, yield algin, and the carrageenan, it is obtained from the red algae, that is Chondrus crispus. Agar extracted from the red algae, that is Gelidium and Gracilaria, is used in the microbial culture, and is also used in preparation of the ice creams and jellies, right? So it gives you the jelly-like combinations. Chlorella is an edible and used in the space shuttle, and it is also employed for the extraction of antibiotics. So this is used, it is edible alga and it is used in the space shuttle because it is eaten by the person who goes to the space with the spacecraft, right? And it also used for the extraction of antibiotics. Brown algae, laminaria and macrocystis have antibiotic properties. Fucus and laminaria are rich source of iodine. So iodine are extracted from these fucus and laminaria. Okay, let us discuss some of the question that has been asked in the NEET examination. So, which of the following is incorrectly matched? Eulothrix, mannitol, porphyra, Floridian starch, volvox, starch, ectocarpus, fucoxanthin. So, ectocarpus, it is having the fucoxanthin, and volvox, it is green algae, they are having the starch as a deposition food store. Porphyra, that is having Floridian starch, red algae. Eulothrix is a green algae, but mannitol, it is a stored food material for the brown algae. So this is not matching. So this is the correct incorrect match. Next question. Which of the following pairs include the unicellular algae? Gelidium and Gracilaria, Anabina, Volvox, Chlorella, Spirulina, Laminaria, Sargassum. So Laminaria and Sargassum, they are not unicellular, they are quite big algae and they are present in the marine environment. Chlorella and Spirulina, these both are unicellular. Anabina is filamentous, unicellular, and Volvox, it is a colonial form. So this is not the correct answer, and this is also not the correct answer. Chlorella and Spirulina is the correct answer because these two are the unicellular algae. Next question. An example of colonial alga: Volvox, Eulothrix, Spirogyra, Chlorella. So chlorella is a unicellular form, Spirogyra is a green algae that is filamentous, and Eulothrix is also filamentous. But Volvox is a colonial form of green algae. An example of colonial algae, that is Volvox. Next question. Which of the following statement is wrong? Algae increases the level of dissolved oxygen in the immediate environment. Algin is obtained from the red algae and carrageenan from the brown algae. Agar is obtained from the Gelidium and Gracilaria. Laminaria and Sargassum are used as a food. So first one is correct. Algae is increase the level of dissolved oxygen because they are doing the process of photosynthesis. So it increases the dissolved oxygen in water. Let us discuss the third one first. Agar and agar is obtained from the Gelidium and Gracilaria red algae. These are the two red algae that gives you this agar and agar. Laminaria and Sargassum are used as a food material. These are the two large algae that are used as a food material. This algin is obtained from the brown alga, right? And carrageenan is obtained from the red alga. So this is the incorrect statement, right? So this is the wrong statement. Last question. Hydrocolloid carrageen is obtained from the which one? Phaeophyceae, Rhodophyceae, only Rhodophyceae, only Phaeophyceae, Chlorophyceae and Phaeophyceae. So first thing, Chlorophyceae do not gives you the any hydrocolloid, whereas the Phaeophyceae and Rhodophyceae both gives you the hydrocolloid carrageen. So this is the first one. This is the first one, that is the hydrocolloid carrageen is obtained from the Phaeophyceae and the Rhodophyceae. So this is all about this session. We have discussed about the algae, right? And in the next session, we are going to study about the bryophyte, pteridophyte, gymnosperm, and small basis of the angiosperm, because angiosperm we are going to discuss in the great detail in other chapters also. Thank you very much.